Hello, and a very warm welcome to everyone to our today's webinar on challenges and consideration in designing and conducting phase 2 Immuno Oncology clinical trial. I am Anvesha Bhattacharya and I represent Veeda Clinical Research. Veeda Clinical Research, together with its subsidiary, Bioneeds India Private Limited, and its joint venture, Ingenuity Biosciences Private Limited, together referred to as Veeda Group, offers a comprehensive portfolio of clinical, preclinical, bioanalytical services to support innovative biosimilar and generic drug development programs of our global clientele. Together, we serve clients globally in pharma and biopharmaceutical, agrochemical and industrial chemicals, herbal nutraceuticals, medical devices industries. So as a facilitator today, I would like to introduce you all to a couple of points as we begin this session. This session will have presentation in the beginning, followed by Q&A round. We have enabled our Ask a Question feature. It's right on the top of your screen. Drop all your questions related to this webinar right there. Now moving forward, I would like to introduce you all to our speakers and panelists for today's session. So our first speaker, Dr. Ravi Alamchandani, with over 6 years of experience in the field of medical and safety reporting. His experience includes working in various hospitals, pharmaceutical companies, and contract research organizations like DDMM Heart Institute, Torrent Pharmaceuticals, Lambda Therapeutic Research, before joining Veeda Clinical Research. He is currently associated with Veeda as a leading team involved in medical monitoring, medical writing, and pharmacovigilance. He has been involved in various teams managing in medical writing and medical monitoring, reporting activities of phase 1 to phase 4 clinical studies, efficacy studies involving NCE, biologics, and generics. Our next speaker is Dr. Ashutosh Jani. With over 17 years of experience in the field of clinical research, he has experience in working academia, pharmaceutical companies, and contract research organizations like Claris, Acutis Research, and Lambda Therapeutic Research before joining Veeda. During his tenure, he has managed teams involved in clinical operations and BD and is having good rich experience in clinical development, product management, contract research organizations, contract negotiations, implementing of programs in therapeutic areas like oncology, psychiatry, endocrinology, inflammatory diseases, including multiple rare and complex indications. Our next speaker, Mr. Jaimin Vaishnav, currently working as AGM in Veeda in project management team. He has 13 plus years of experience in the field of project management. He has worked in various pharmaceutical companies and contract research organizations, including Torrent Pharma, Intas Pharma, Clienta Research, having area of expertise in handling IND, NCEs, biosimilar drugs, complex generics in oncology and psychiatry as a therapeutic area. So without any further delay, I would like to call upon Dr. Ravi to begin this session.
Thanks for the introduction, Anvesha. We can start with the slide deck. So we'll be discussing all the challenges and what all we consider while designing or conducting phase 2 immuno-oncology trials. Now, before we get into the nitty-gritty of clinical trials of these immuno-oncology products, we first need to understand what are these immuno-oncology products per se. Next. Next. So these are the sections that we'll be covering today. So we are all aware what is immunotherapy. It's basically the body's immune defense, which helps us fight various diseases. What is immuno-oncology? Now, usually our immune system is capable of fighting even the abnormal cells in the body. And these cells that can develop into cancerous cells are usually killed by our defense system. But sometimes these cancer cells mutate to an extent that they escape our immune system. And in this case, they become threat to our health and can grow into tumors. Immunotherapy that is specifically targeted to cancer is something called as immuno-oncology. Herein, the body's natural defense is used to fight cancer. The immune system is modified and stimulated in such a way that it is able to now fight cancer that it is not naturally able to do so. Lymphocytes, which play an important role in our immune system, are genetically modified and mobilized to eliminate the cancer cells while in immuno-oncology. Next. Now, what are the advantages of this immuno-oncology or this immune therapy, I would call it, over the conventional treatment? The conventional treatments are very non-specific and they do as much harm to the patients as they do good. They not only kill the cancerous cells, but they also kill the healthy cells in the body. Or sometimes it is not possible for these conventional therapies to kill all the cancerous cells. On the other hand, the immuno-oncology or the immune therapy in oncology category is targeted specifically at the cancer cells that have particular antigen, particular receptor, or so on. And immuno-oncology can be targeted at all the stages of the tumor. There is no restriction as to whether you treat stage 1 cancers or stage 4 cancers. You can target all the stages of cancer with immune therapy. And since this is the immune system, the body has a tendency to produce memory cells. Thus, the memory stays for long and the chances of recurrence are less. The immune therapy in oncology indication is in existence for about more than a decade now, with first approval being received in the year 2010 for treating melanoma. Next. The immuno-oncology therapies can be categorized as specific or non-specific. As specific like vaccines, antibodies, non-specific like cytokine inhibitors, like antibodies against the checkpoint inhibitors. Active as well as passive. Active, which directs the immune system to attack the antigens like vaccines or adaptive cellular therapies like CAR-T therapy. Or these are categorized as passive, which basically enhance the tumor response. The tumors get altered in a way that they become more reachable to the immune system, like antibodies against the checkpoint inhibitors and cytokine inhibitors. Next. These are the various immuno-oncology products, adaptive cell therapies like CAR-T therapy, dendritic cells, antibodies against checkpoint inhibitors, against bispecific T cell engaging, then cytokines, vaccines, oncolytic viruses, or a combination of these all. These are the different immune oncology products. Next. Now discussing few of the immune oncology therapies that are currently approved as well as are being developed for other cancers. One such cancer therapy or the immune oncology therapy is chimeric antigen receptor T cell therapy or the CAR-T therapy. It's a genetically modified immuno-oncology therapy wherein the patient's T cells are removed from their body and they are re-engineered in the lab to produce CARs, that is chimeric antigen receptors on the surface of these T cells. These modified T cells are now known as CAR-T cells. Now, before administering these back into the patient's body, the patients are lymphodepleted, that is, their existing lymphocytes are killed using a short course of chemotherapy. Subsequent to that, the CAR-T cells are injected into the patient. And these CAR-T cells, they ultimately attack the cancerous cell. Now, CAR-T cells are active in the body for a very long period, and thus they prevent recurrence of the cancer for a long time. And patients tend to be in remission for a longer period. In late 2017, FDA gave approval to Novartis CAR-T therapy for treatment of acute lymphoblastic leukemia in children. Kite Pharma is developing CAR-T therapy for management of aggressive B-cell non-Hodgkin lymphoma in adults. Biotech with Clengene is developing CAR-T therapy to fight multiple myeloma in early stage, and this development is in early stages. So we can see that various organizations these days are developing CAR-T therapies for fighting the cancers. Next. Then monoclonal antibodies. These monoclonal antibodies can be directed towards the antigen, or these can be directed towards the checkpoint inhibitors. Now, towards the antigen, we almost are well aware of trastuzumab being used in various cancers, which blocks the hERG receptors, and we are also aware of altuzumab. This therapy basically This therapy is basically a use of antibodies, the genetically engineered antibodies that are targeted towards a specific antigen of the tumor. They bind to the tumor and they trigger an immune response, which ultimately results into killing of these cancer cells. The next category of monoclonal antibodies are directed against the checkpoint inhibitors. Now, in the human body, there are various checkpoint inhibitors. There are various inhibitors which they bind to certain checkpoint proteins. And when this binding occurs, say binding between PD-1 protein and another protein called PD-L1, when this binding occurs, the immune system is prevented from attacking the body cells. It so happens that some cancers, they develop so much abundant PD-L1 protein that this PD-L1 protein, it coats the PD-1 protein, and this prevents the attack of the immune system on these cancers. So these cancer cells, they tend to grow as normal cells in the body since they have hidden themselves from the immune system. Monoclonal antibodies have been developed which can block the binding of PD-L1 protein with the PD-1 protein, thus exposing the cancer cells to the immune system and thereby the immune system killing these cancer cells. Some of the antibodies or the therapies that have been developed to target PD-1 receptors are ipilimumab, which was the first drug to receive approval way back in 2010, nivolumab, and pembrolizumab, which is the most recent one, which is being used to fight various cancers like melanoma, lung cancer, and so on. However, sometimes these antibodies can trigger serious adverse event or severe immune-related adverse event, which we will be discussing subsequently. Next slide. Next. Now, cytokines. Cytokines are basically the part of immune system in the body that help to maintain the balance in the immune system. If this balance of the cytokines is impaired by their increased production or decreased production, there may be abnormal cell growth, which can result into cancer. JAK family of proteins are crucial cytokines. Excessive JAK proteins, they result into various inflammatory diseases as well as certain cancers. Inhibition of these JAK family of proteins can help treat not only cancers, but also certain autoimmune diseases like rheumatoid arthritis, psoriasis, and alopecia. Next. We have also been successful in developing vaccines that target or that boost the immune system to target the cancer cells. Now, these vaccines may be made up of different components of cancer cells, like tumor antigens, cancer cell DNAs, or these will non-specifically stimulate the immune cells. Now, these vaccines activate the patient's immune system, which then attacks the cancer cells and kills them. Again, these are vaccines. They tend to create memory cells. They are there in the body for long. So recurrence are less and remission is for a longer duration. Next. So that's grossly about the immuno-oncology therapies currently available in the market or are being developed. My colleague Jamin will take forward from here. Thank you.
Yeah. So thank you, Dr. Ravi, for wonderful insight on the immuno-oncology product. Next slide, please. Yeah. So we start with the part 2 of the presentation with the quote from the famous Kung Fu master, Master Oogway, that there is always something more to learn, even for a master. So our contention here is to learn each day. And if you read an article each day, so obviously it will be like 300 articles a day you have a reading. So we all learn, and that's why we are here. So thank you so much for your time. Next. So now we go for the consideration for the clinical trial. So if we can divide into the three phases, one is the study conduct, the second is for the study design, and the third is the regulatory approval. In the study conduct phase, we are going to discuss with the patient eligibility criteria, product collection, adverse event is diagnosis, grading and the management, response assessment, and the patient engagement. Yeah. Next. So we start with the study design part. So the traditional clinical trial for the cytotoxic drugs, so that I have always followed phase 1, phase 2, and phase 3 intervention clinical trial with 3+3 design with the dose escalation and the dose expansion across the various stages of the clinical trial. So we all know that the first 3 patients will be exposed to one particular dose, and based on that response or the dose limiting toxicity or the maximum tolerated dose, the further 3 patients will be have the dose escalation or the dose de-escalation. Now, if we consider the development of the immuno-oncology clinical trial, so it is very critical to identify the right dose in the early phase of the clinical trial, so that improve the probability of the success in the late phase clinical trial. We all know that immuno-oncology products have complex mechanism of action, as Dr. Ravi has already explained. So how they are different compared to the traditional cytotoxic drug? So they do not act directly on the malignant cells. They act on the immune system. They strengthen the immune system, and then you will get the response. So we can see the traditional, the monotone assumption that you increase the dose and you get the more response, and then there will be the more kind of toxicity. So that will not work for the immuno-oncology product. So although they have the same Cmax and the AUC, but their PK and PD do not require to be matched. So that is a main limitation for the development or the clinical trial of the immuno-oncology product. So we have put one example here. For example, PD1 receptor occupancy. So that is a median occupancy that is 64 to 70%. But if you consider the dose level, so the dose level range from 0.1 to 10 mg per kg. So even if you increase the dose level several fold for 8 times to, I can say, the 100 times, the receptor occupancy is not changing them much. So it is we need to consider this point for the clinical trial of the immuno-oncology drug. Next. So as you can see, now the concept of the master protocol that has been the famous across the field of the clinical trial. Master protocol can be divided into the 3 parts. So the basket trial, the umbrella trial, and the platform trial. We start with the basket trial. So basket trial refer to a design where the targeted therapy is evaluated in the multiple diseases, and then that have the common molecular alternation. So that is the basket trial. Umbrella trial, on the other hand, have the single disease, and then you have the multiple intervention 1, 2, and 3. So that's the umbrella trial. The platform trial that also considered as the multiple stage and the multiple dose trial. So in that case, if you can see that there are different arms, different intervention will be evaluated against the standard of care or the reference product. The advantage of the platform trial is you can, this is a flexible design trial, and we can, the arm that is not working, so we can drop that arm and we can add the new arm. So if you can see the part 2 of the interim analysis, so the intervention 1 is not working, so that arm is particularly dropped, and then we have introduced intervention 3. Again, we have introduced new arm, that is intervention 4, and then we can have the final analysis. The landmark approval of pembrolizumab by the FDA, where the phase 1 trial was initiated in year 2011, and the drug was approved by the FDA in 2014. So now the industry and the CRO that focus on the master trial for development of the master protocol that on the development of the immuno-oncology product. Next. So as you can see, there is a rapid change in the master protocol. So the concept is being used, and now if you can see, so we have the multiple of the trial using the same study design. So this point we need to consider during the clinical trial of the immuno-oncology product. Next. Now we move to the study conduct phase. So for any of the study conduct, and especially for the phase 2, so we can that rely on the multiple factor. So the first thought that will come, that is for the site selection, the patient selection, biomarker that can help us to identify the response, product collection and the tracking, and the response assessment. Next. So on which base we are selecting the site. So the first so there are multiple factors. We have listed down a few of the factors that can play a key role for the study conduct during the phase 2. So the first is the previous similar clinical trial experience. So the site know how to handle the immuno-oncology product and also to conduct the clinical trial. The second is the experience and the trained sites team. So because you need to have the product collected, so obviously you need a trained team member. The third is the site monitoring team. So this is a new concept where the site will have their own QC team that oversee the development and the conduct of the clinical trial. The next is the PA training on the immune-related adverse event. We all know that there are the several adverse events that is particularly useful to understand for the PA regarding the immune-related adverse event. So that also we check during the selection of the site. The next is the site infrastructure. So it's mainly useful for the stem cell therapy or even the CAR T therapy, where the product is, the patient will be enrolled and then you collect the T cell, then you have the re-engineer them and then you infuse into the patient. So the site infrastructure that also play a key role during the conduct and the selection of this site. The next is the patient coordination. So this is the most crucial part in all the clinical trial. Patient, we need a more experience in the trained site team that help us for the patient coordination, patient visit schedule. If they require, we can have the patient travel support also. It is approved in the ICF and by the HS committee. The next is the site accreditation. So obviously site accreditation play a crucial role where the regulator have the assurance that the trial is being conducted as per the ICGCP or the FDA or the EMA standard. And the last is the PI oversight on the overall conduct of the clinical trial. So based on all the CRO and have the database. So we consider this factor for selection of the site for the conduct of the phase 2 immuno-oncology product. Next. Now regarding the patient selection. We all have the patient that are coming to site. So we first select the tumor type. So which tumor we want to be targeted or which tumor aim to be studied, that we need to identify. And many a time we know that the collaboration with the central lab or the central kind of lab, so that will help us to identify the patient pool, where the patients are coming from. So understanding of the tumor type, that also help us to understand the patient selection. The next is the tumor biomarker. Which biomarker we are targeting, whether it's a diagnostic or the predictive biomarker. So for example, the PD-L1 expression can help us to predict the patient response. The next is the disease stage. If the patient are coming with the metastatic disease, so already they have been exposed to the prior chemotherapy. So in that scenario, the patient are eligible for the second line or the later treatment phase of the IO product. And if the patient have the earlier stage disease, then we are looking for the neoadjuvant or the adjuvant treatment. The next is the prior treatment. So the type and the number of the prior cancer treatment that the patient have received, that may impact their eligibility in the immuno-oncology trial. And the last is the patient health. So the patient with the underlying comorbid condition, including the autoimmune disorder, so may not be eligible for the immuno-oncology product. Next. So we continue with the patient population and the eligibility. So now if we are looking for the phase 2 trial for the conduct, so the patient participation, that is a crucial factor we need to consider, because this is a new therapy we are bringing into the market. So patient always have the reservation that this therapy will work for me or not. And obviously we are looking for the targeted therapy. So the patient participation, patient encouragement, that is a crucial part for the PI and for the site team. The next is we need to design the inclusion and exclusion criteria. So that will help us, that improve the odds of success for to meet the primary and the secondary endpoint. The next is to, if we have the confusion of any of the inclusion and exclusion criteria, then we can take help of the medical monitor to verify the patient eligibility. So we ensure that the right patient have been enrolled into the clinical trial. The next is exposure to the other immuno-oncology product over the last 6 months or the 1 year. Why we are looking for the 6 months or the 1 year? Because we know that the standard of care that have 3 to 6 cycle based on the treatment regime. So we are looking for the patient who have the exposure of the previous immuno-oncology product. The next is the patient condition. So we are looking for the patient who have the comorbid condition, then patient age, ECOG status. So many of the trial, if you look into the inclusion and exclusion criteria, that only allow the patient with ECOG status 0 or 1, then any of the significant disease history. So we are not allowing this kind of patient into the trial based on the inclusion and exclusion criteria. The next is the concurrent therapeutic or the supportive care management. If anything will go wrong or the patient have the adverse event, so are we providing any standard of care? So that also will affect our patient recruitment and the continuation in the clinical trial. And the last one is the pediatric versus adult population. Our targeted intervention will work in the pediatric population or the adult population. So that also will a key factor to consider in the conduct of the clinical trial. Next. So this is all about the heterogeneity of the receptor type, how it has been evolved. So if you know the NCCN guidelines, so in the early 90s, early 80s, it is not like we have the receptor phenotyping is available. But as you can see in the breast cancer, we have now the different type of cancer receptor that is available for distinct, but is for the HR positive negative or the ER negative positive or the HER2 or even for the triple negative breast cancer. So we need to consider the patient heterogeneity during the designing and the conduct of the clinical trial. And that will definitely going to a big factor to consider for the meeting for the primary or the secondary endpoint. Next. Patient selection have the multiple, the criteria. So regarding the procedure, if you can see how it has been moved for the procedure for the inclusion and exclusion criteria, different countries, and how the landscape is changing over the year regarding number of site and the number of patient. Next. The next is the product collection and the tracking. For the stem cell therapy or CAR T therapy, this is the most important part during the study conduct. So for where the central lab is situated, that is the most important part. So the turnaround time for the laboratory to consider the key decision. So you have collected a patient T cell or any of the cell, and then you have extracted, you have stored at your site based on the coordination with the central lab. You have shipped all the sample to central lab. So they need to get re-engineer, and then again the cell will come to your site back for the patient treatment. So we need to consider all turnaround time, and obviously that is a key factor for the success of the clinical trial. We need to understand the sample logistic, and we understand the key risk, and we need to have the mitigation plan in place for all the key risks that we have identified. The next step is the coordination with the patient. If the patient is coming from the remote area, or because for the onco patient, obviously you will need a kind of attendee with them for their travel. So that also play a role in the patient product collection, and if required, so we can also provide the patient travel support as per approved by the ICF and in the HS committee. The next is a specific technique for the sample collection. So we're looking for the targeted therapy. So we will need a trained principal investigator and the site team. So they are clearly educated and trained by with all the aspect of the sample collection. Now, if we move to the laboratory requirement for the sample collection, the processing, storage at the site, we say the cryopreservation, the shipment to the central lab, and when the patient actually receives the treatment. So this is all around the turnaround time. So we wish that all this turnaround time should be the minimal one, and the patient should receive the treatment as early as possible. Next. Okay. The biomarker in immuno-oncology. So biomarker is a measurable biological factor that can provide insight to each patient, individual cancer, and which doctor can use to determine the approach most likely to benefit a particular person. So we are looking for three types of biomarker. One is a predictive biomarker. So this kind of biomarker help us, to assess whether individual will respond to treatment. Let's say, for example, the HER2 positive or the ERPR positive or the therapies that have already been approved, that is trastuzumab, pertuzumab. These are therapy already approved based on the predictive biomarker. So you know that the assess this biomarker and the response will be assured. The pharmacodynamic biomarker that show that the biological response has already been occurred, and that that will help us to take the key decision. And the third is the prognostic biomarker that identify the likelihood of a clinical event. So especially for the AML patient, when you understand the specific gene mutation, so that will identify the next key event. The biomarker are the PD-L1 expression, then pre-existing immunity, tumor genomic instability, and the microbiome. Next. Yeah, thank you. So I handed over the presentation to Dr. Ashutosh Jani. Thank you.
Okay, thank you, Jamin, for setting up the stage with respect to the biomarkers. I'm not able to switch on the video. Yeah. Okay. So thank you, Jamin, for setting up the stage with respect to the biomarkers, and the biomarkers are definitely an integral part of any immuno-oncology trial which we conduct, especially if we categorize the biomarkers identification. We could say like based on the tumor tissue analysis of that. So one of the most common ways in which we identify biomarkers in immuno-oncology trials is through the analysis of the tumor tissue, which we typically get it from the patients. And this can involve examining the specific genes, proteins, or immune cells, any markers, using different techniques, including the immunohistochemistry. So gene expression and then profiling, or through flow cytometry, we get more insight about the disease per se, and we are able to evaluate the disease. The others are the blood-based biomarkers. So in addition to the analysis of the tumor tissue, some immuno-oncology trials may also use blood as a basis of biomarker to monitor the patient's response to the therapy. And these biomarkers can include either circulating tumor DNAs or the tumor-derived exosomes, or specific immune cells subsets, especially in the blood stream. The third and important aspect is imaging-based biomarkers, which typically use it for many of the oncology trials. So imaging techniques has PET, like say we use the positive emission tomography or magnetic resonance, that's MRI, And with these instruments, we try to identify the immuno trials biomarkers, for example, tumor size or the metabolic activity on the PET scan or the image, or the patient's response to therapy could be evaluated using these imaging techniques. The last and the recent one is typically now we have machine learning algorithms. So these machine learning algorithms can be used to analyze large data set of the patient's information. So we feed in all the patient related information, and that could give you a predictive analysis of the possibility of the patient getting into a tumor, or the possibility of having breast cancer or any type of oncology. This is based on the data of the family history and all the patient data available. We can go to the next slide. So the biomarkers, as we mentioned, like say the PD1, PDL1, that is the programmed death ligand 1. So this is typically a checkpoint which shutdowns this anti-tumor natural killer cells. And the blocking of this programmed death 1 or programmed death ligand 1 pathway can overcome these expressions. And the FDA has approved this 7 checkpoint inhibitors like this for the treatment of various solid as well as hematological cancers. The other is, like say, the marker could be the pre-existing immunity. So the tumor already infiltrated by the natural killer cells. So we can count the CD8 or CD4 plus cells and get a fair idea about the biomarker for the immuno-oncology studies. Another is the tumor mutational burden. So the tumor mutational burden is dependent on the number of somatic mutations in the tumor genome. And this mutation typically fuels the cancer, but also makes the cancer stand out of the immune system. And this could be used as an evaluation parameter. So heavily mutated tumors are more likely to respond to immunotherapy. And the FDA has approved NSG panels for the use to estimate this tumor mutational burdens. Another is the microbiome. So there are trillions of bacteria inside our intestine, and the higher gut diversity associated with the higher response rates after the anti-PD1 or PDL1 checkpoint therapy. And antibiotic use, rampant use is also associated with worsening of the survival of this checkpoint immunotherapy. Go to the next slide. The other, as we saw through the design aspect from Jamin, so the other endpoint designs which typically are used for immuno-oncology products are the usual overall survival. So that is clinical endpoint or the gold standard for typical oncology clinical trials. This typically doesn't give always a practical use practically because of the large patient numbers are needed for this clinical endpoint. The other is the progression-free survival. So this is measures of the time from the treatment initiation to the beginning of the disease progression. So until what time the patient is remaining free of the disease is what we measure. Most commonly used for oncology studies. And overall response rate or ORR, that is the proportion of patients with reduced tumor burden by predefined amount. So how much tumor are we able to reduce? We try to see that through an evaluation parameter with RECIST. So we try to see whether there's a complete response, partial response, the disease is stable, or there is a progression in the disease. Move to the next slide. So the imaging endpoints for immuno-oncology are slightly different. So the trial depends on the imaging endpoints to predict the survival. However, without accurate imaging endpoints of the response to immuno-oncology, successful trial would not be possible. And radiographic response to immuno-oncology are unlike response to chemotherapy, where the tumor cell killing, the shrinkage are more immediate. And the flare effect of immuno-oncology describes the initial tumor burden typically increases in the size and then followed by delayed response and then a shrinkage. So that's the typical pattern in which immuno-oncology therapy works. You can go to the next slide. So because of this different nature in which the immuno-oncotherapy works, we can use this imaging techniques like CT scan or MRI scan or FDG, 18 FDG PET scan, etc. for a bone scan or chest X-ray. These are the imaging techniques which are typically used for the evaluation of the tumor size. Yeah, go to the next slide. So as we saw through the imaging response criteria, which we see, the immunotherapies may be used in multiple clinical settings, like say either it could be adjuvant, first line, second line, or subsequent line of therapy. The FDA guidance on oncology imaging typically uses RECIST 1.1. Now the design for randomized studies, planning for licensing application continue to use the RECIST 1.1 as a primary criteria, as an endpoint criteria. But RECIST 1.1 might not be adequately capture the unique pattern which we typically saw in case of immuno-oncology drugs. So the trials with immuno-oncology use because typically use a combination of standard therapies, that is the chemotherapy as well as radiation, some novel entities, and further compound the assessment of progression based on the endpoint. So iRECIST, which is typically used for immunotherapies, should be regarded as exploratory and could be used for immuno-oncology type of therapies. We can go to the next slide. So the RECIST typically, if we see, is used more for solid tumors, and it's been established way in 2000, and there's a RECIST 1.1 that's upgraded version in 2009. They typically measure the tumor burden based on the sum of diameter of the tumor, is how typically the RECIST works. Go to the next slide. However, as we saw through that in immuno-oncology, the RECIST may not be of that usefulness. Because of that, we can use this iRECIST, which was published by the same RECIST group in 2017. So the RECIST 1 and 1.1 has this limitation in assessing the response, especially with respect to immuno-oncology therapies. So the working group recommends use of the new standard terminologies as immune complete response, or immune stable disease, or immune partial response, or immune unconfirmed progressive disease, or immune confirmed progressive disease. So these are the parameters through which we evaluate immuno-oncology drugs using iRECIST. And the key change typically when we give immuno-oncology drugs is that there's a flare effect which needs to be taken into account. So the definition of the rule for confirmation of progression of the disease. So these new parameters of IUPD, that is the immune unconfirmed progression of the disease, is used. Now, IUPD is confirmed only after 4 to 8 weeks. You can go to the next slide. So this typically we try to see through the difference between RECIST and iRECIST. So majority of the parameters between RECIST and iRECIST remains the same. But typically the immuno-oncology products, the response is delayed, and there is an initial flare, as we already seen in the disease subsequent to the immunotherapy. The criteria for this iRECIST is IUPD, and this needs to be evaluated or confirmed only after nearly about 4 to 8 weeks of the treatment. We're able to derive the IUPD. We can go to the next slide. As we see through the RECIST, and we typically use iRECIST. Similarly, when we need to check on for adverse event, there are high probability of chances that the adverse event may be because of the immunotherapy which has been administered. So we differentiate the adverse event as IR adverse events or the immune related adverse event of the early or the late onset. The early onset of IRAs could be inflammatory reaction to the immunotherapy treatment, such as either hepatitis, enterocolitis, hypophytis, nephritis. So these are typically the A's which could be related because of the immunotherapy treatment which has been given. Among the late onset are mostly the common fatigue, extensive rash or itching, diarrhea with blood or mucus, or severe abdominal pain, weight loss, nausea, vomiting. So these are all delayed adverse event which could be seen in patients administered immuno-oncotherapy. The late onset adverse event could occur as late as about 10 to 16 weeks after the treatment initiation. And in this context, the observation of DLT and the late onset of IRAs is what is need to be considered. The predictive biomarker for the immune related adverse event is not yet typically defined. We can go to the next slide. So just seeing on to the general adverse event which are seen, especially in case of immuno-therapies, there's the cytokine release syndrome, that is the CRS. So there's a systemic inflammatory response which is characterized by fever, hypotension, tachycardia, tachypnea, hypoxia due to supraphysiological cytokine elevation. So these are cytokine release syndromes. The others are immune effector cells or associated with neurotoxicity syndrome. So there is a constellation of enterocephalic signs and symptoms, and there is a altered level of unconsciousness, impaired cognition, motor weakness, seizures, etc. So we also refer to them as CRES, especially in case of CAR-related encephalopathy syndrome. And due to the cytokine elevation, these immune effector syndromes are seen. The other is the macrophage activation syndrome, that is the systemic inflammation syndrome, where there is adverse event of either fever, splenomegaly, hepatomegaly, elevated levels of ferritin, cytopenia, liver dysfunction, and coagulopathy. So these are typical common adverse events associated with the immunotherapies. You can go to the next slide. So the immune effect cell-associated neurotoxicity syndrome is one of the most common ones. So there is a constellation of encephalopathic signs and symptoms. So as we already see through, there is aphasia, that's altered level of consciousness, impaired cognition, motor weakness, seizures, or there be overlapping of symptoms with other pathophysiological conditions. And the characteristic of the patient who is awake or mute and does not respond verbally or physically to any examination. Also referred to as CRAs due to cytokine elevation. And we can go to the next slide. So these adverse events which we saw through the immune IRAs, there's different checkpoint inhibitors also. There's a clear pattern of IRAs that has been defined in this immune checkpoint inhibitors. And according to the recent review, the overall incidence of life-threatening or severe immune-related adverse event grade 3 ranges from about 20 to 30% of the patient receiving ipilimumab, or from about 10 to 15% of the patient receiving anti-PD-1 agents. So, and it was highest about nearly 55% in patient receiving anti-CLAT or PD-1 combination therapy. So there are high chances of this typical adverse event, and this should be aligned with the mechanism of the action which has been used for targeting these immune effector cells. The IRAs from the anti-CLAT, CLAT4 agents and dose dependent, and occur more frequently in dermatological or gastrointestinal or endocrine system, whereas the anti-PD-1 agent toxicities appears to be more dose dependent. So as we already seen through that, predictive biomarkers for IRAs are not yet defined. We can go to the next slide. So these are the different adverse event also inhibitors which we target. We can go to the next slide. So the treatment of these adverse event or immune adverse events is typically important. So whenever we find that there are IRAs, we first need to hold the medication for a grade 2, more than grade 2 toxicities, and then initiate the steroids and permanently discontinue immune checkpoint inhibitors for if the grade 3 toxicities are higher. Corticosteroids are the mainstay for the therapy for moderate to severe IRAs, and the second line of ICI, that's infliximab or mycophenolate, typically needs to be given. CRS like tocilizumab or cetuximab are also used to treat adverse event arising because of immune related IRAs. You can go to the next slide. Regulatory definitely is a challenging aspect, especially when we come to immuno-oncology type of studies. So regulatory agencies have been under enormous pressure to make the approval process more efficient and less expensive. So oncology developers have benefited from this breakthrough therapy, and the FDA has given approval to nearly about 16 such drug design breakthrough therapies. Of the 11 were the first time approval for the novel drug, so which includes pembrolizumab. So pembrolizumab has been a breakthrough therapy, and it's been we've seen through the master protocol. So this pembrolizumab was one of the key drug which was being evaluated through this master protocol, and the approval timelines was the quickest as far as I remember. And the combination for better trial design and the possibility to break through the design, which will be allowing the companies to conduct small clinical trials, which will be faster in small group of patients, and which lead to accurate or accelerated approvals in the niche market, is what is the need of the hour. If we consider India, so in India, I think we don't have any CAR-T or any immune type therapy, which has been CAR-T type of therapy, which has been yet approved by the regulators. Yeah, we can go to the next slide. So in summary, multiple biomarker assays are available for assessment of efficacy of immuno-oncology therapies, hence the selection of biomarkers will be a challenge. And the rapid changing standard of care can lead to change in the study design and the conduct. And highly complex studies and derivatives of RP2D is there. The new safety signal or the adverse event and combination therapies require the PI training along with experienced and trained monitors that is needed, and with thoughtful consideration of critical factors during the study design and conduct will lead to further drug development accelerate of the new immuno-oncology medicines to the patient. So that's what in a nutshell we summarize based on the current webinar. Yeah, I think we go to the next slide. Yeah, so I think we are open to questions.
Thank you, Dr. Ravi. Thank you, Dr. Ashutosh. Thank you, Jamin, for such wonderful presentation. Now moving on, we will be proceeding.
Yes, Anvesha, if we have any questions, we can address the same.
Yes, so we will be proceeding ahead our discussion and some of the questions posed by our audience. So my first question to Dr. Ashutosh is, how challenging is it to conduct phase 2 clinical trials?
Yeah, Anvesha, so as we saw through the presentation as depicted by Jamin, so there are definitely challenges when we come to phase 2 clinical trials, especially it starts with the designing and further evaluation of the safety and efficacy of these new treatments after the initial safety has been established in the typical phase 1. So, however, there are several challenges when we get into the phase 2. So, patient recruitment is a challenge. So, recruiting enough patients who meet the eligibility criteria for the trial can be a significant challenge, particularly if the patient population is rare and the trial which requires specific subset of the patient, as we see through, like say, triple negative breast cancer or HER2 positive, etc. The sample size determination is also a challenge because we are dealing with like determining the appropriate number of patients for a phase 2 trial can be challenging, especially it requires balancing the power with the need to minimize the patient exposure to potentially ineffective or harmful treatment. With very limited data available probably from a phase 1 studies, it's typically difficult to estimate the exact number of patients needed for a typical phase 2 study. The another aspect at times is blinding, especially maintaining blinding in a phase 2 clinical trial can be challenging, particularly in trials where it involves multiple treatment arm, as we saw by Jamin, that there are different various designs like a platform design or the adaptive designs which we have seen through. So there were multiple arms which would be there, and the treatment has noticeable effect that patients or the researchers can easily identify. So in these situations, maintaining a blind, especially in a phase 2 type of immuno-oncology study, is definitely challenging. Yeah.
All right. Thank you, Dr. Ashutosh. My next question to Jamin is, does Veeda have experience with adaptive study designs?
Yes, so Veeda have conducted one phase 2 study design with the adaptive design for the COVID-19 study, where we have recruited 1,500 patients. So yes, we have the capability of to have the adaptive study design, including the seamless phase 2 and the phase 3 clinical study. Thank you.
All right. Thank you, Jamin. My last question to you is, are there any indigenous card available in India?
Yes, so if this is still under development, that is not been approved, but yes, we have the Tata Medical Hospital Mumbai, and we have the Department of the India. So they have developed a one CAR T therapy that is available. Phase 1 is already been completed, and we are moving for the phase 2. So hopefully that will get into phase 2 and phase 3, and maybe patient have access to that medication soon in India. And the cost of the development of that treatment is very less compared to the Western countries. And obviously, ultimately, the cost to patient also will be much minimal. Thank you.
All right. Thank you, Jamin. So I would like to thank all the speakers for nicely answering most of our audience questions. I would like to take a pause over here. We understand this session has kept us well engaged throughout, but due to some time constraint, we would not be able to take any further questions at this point of time. But please be rest assured that we will be sending the answers to every questions of yours via emails. Also, we will be sending across the recorded link to the webinar on all registered email IDs. Lastly, I, on behalf of Veeda Clinical Research, would like to sincerely thank you all for being here today and taking out the time to be part of this webinar. I would also like to extend our gratitude to all the speakers for being such wonderful presenters and giving us all great learnings today. We at Veeda Clinical Research earnestly look forward to hosting you all again. Till then, take care and stay safe, everyone. Thank you.